I'm going to start with the good news. You're all going to live really long. That's at least in historical terms and statistically speaking. Here's an amazing statistic for you. This is how human life expectancy has developed since the Industrial Revolution. In 1840, human life expectancy was 45 years. And human life expectancy ever since then has seen a massive increase that's really been unprecedented in human history. And if you think it's a very short piece of human history, such that human life expectancy is 80 years in age in the 2000s. So you might truly ask, why is it that we study aging in the first place? Is it not obviously a problem that's sort of going away by itself? This trend really doesn't appear to end very soon. Well, I would disagree with that because aging is a major risk factor for diseases. And these are important diseases such as cancer and dementia. And if you think about it, cancer and dementia, they really don't have much in common if you think about the biology of these two diseases, besides the fact that they occur in old people. So this is sort of what biology has in store for all of us, and that's really the bad news. In fact, now it is 1 in 3 persons who dies with Alzheimer's disease. And this is, of course, not only tragic to these individuals, but also a big problem for their families and actually for society as a whole. So when we do aging research, it's really not only about finding ways to reach extreme longevity, but what it's really about is to find ways for healthy aging. So we don't want to produce super long longevity, but we want to really counter and understand age-related diseases and what it is that these diseases have in common. Now, here's a number for you, just to give you a rough idea about the scope of the problem that we're up against. This is the number of DNA lesions that occurs in each person during 20 minutes. So in other words, this is the number of DNA lesions that occurs in each of you during this talk. And yeah, they would also occur if I didn't speak. Now, cells have a whole army of molecular maintenance machines that have the job of fixing these DNA damage events all the time. And really, if these molecular maintenance machines would not work properly, we would be in great trouble. And actually, a decline in this quality control of our genome, of our genes, can lead to diseases of the genome, such as cancer. Here's a bigger number for you. This is the number of proteins that is being synthesized in everybody during the same 20-minute time span. Now, proteins are really amazing structures because really proteins are involved in pretty much every biological process in cells. So there are proteins that are motor proteins, and other proteins actually receive information and transmit information. Even though they have this diverse function, all proteins start out from a very simple structure. And they're really quite amazing because all proteins are really made up of strands of amino acids. And only when proteins fold properly, they can fulfill their individual functions. And protein folding is super complex. This is what a folded protein looks like. And only if this amino acid strand really folds properly can they fulfill the function that each protein has to fulfill. Now, protein folding is a little bit like origami, because also with origami you start from a simple structure, and when you're done with the folding, you end up with a beautiful three-dimensional and complex structure. And just like with origami, when you fold paper, you might be tempted to use a little bit of glue to make things stick. That's also how biology solves parts of the protein folding problem. Actually, there are glue molecules in the cells, little sugar molecules that are added to proteins to help them fold and to help them stay folded. And this is a process that's called glycosylation. And besides glycosylation, we also have a whole host of, again, molecular maintenance mechanisms in place that help proteins fold and stay folded. And again, just like with the genome, the defectiveness of these molecular maintenance mechanisms can lead to diseases. Actually, 30% of all proteins never properly fold because this is such a complex process. And so these unfolded proteins, they're really dangerous for cells because unfolded proteins, they tend to aggregate, and aggregated proteins are toxic to cells. Actually, the diseases, Alzheimer, Parkinson's, Huntington's, all of these diseases can be caused by misfolded and aggregating proteins. Now, As we age, these molecular maintenance mechanisms lose fidelity. As we age, the molecular maintenance that keeps our genes and our genome healthy and our proteins and our proteome healthy to prevent diseases such as cancer and such as dementia, those processes become defective. And this is sort of the underlying reason why age predisposes for these really distinct diseases. In addition to this sort of medical explanation why it's important to study aging, I just want to point out that I think aging is one of the most interesting biological questions that we can ask currently. Because really, if you think about what it is at a molecular level that helps babies, for example, to have this amazing capacity for regeneration, and what it is again at a molecular level that goes away as we all age, is, I think, a very interesting topic, and I think a very interesting piece of biology to study. And I just want to talk a little bit about how we address this problem in the lab. So I told you that protein quality declines with age, and we just wanted to simply ask the question, is the decline in quality control a consequence of aging, or is aging a consequence of the reduced protein quality control? And to do this, we simply asked if we can extend lifespan by improving protein quality. Before I do this, I want to introduce to you the beasts that we're working with in the lab. This is elegans. It's a worm that lives in the soil, and actually here on the screen it looks quite gigantic, but in fact it's only about 1 millimeter in size. And this is one of the reasons why we're using them. We can simply keep lots of them in the lab. At the same time, they don't live very long. They only live for about 3 weeks. And this is the reason why we can easily do survival studies with these animals. And finally, we understand their genetics pretty well, so we can simply and in a straightforward way manipulate their genome. So now you might ask, you're coming here, you want to understand human aging, and you're working on a worm. These worms only have 950 cells after all. But I'm telling you, this is exactly the reason why we're using these animals. We are using simple model organisms in order to understand basic biological problems. Each cell, starting from the simple yeast cell all the way up to our own cells, have to deal with the same biological problems. And those are, for example, how do I keep my genome stable? How do I keep the proteome healthy? By understanding how biology solves these problems in simple model organisms, we think we can learn something about our own biology. So we wanted to use the power of genetics to find new genes that extend lifespan, to find new longevity genes. And in doing that, we can do this in 2 ways. Either we would assume we're smart, or we assume we're not. Assuming that we were smart, we would have developed a hypothesis, which would mean to sit down, read the literature, read the books, and come up with a hypothesis, and then go to the lab and test this hypothesis experimentally. And in this situation, biology typically proves you wrong or right. We didn't assume that we were smart, and what we instead did, we interrogated the whole genome of these worms for mutations that extend lifespan. So what we did is we introduced random mutations across the whole genome in these worms, and then we challenged these mutants that we got out of this. And the challenge was a drug that inhibits protein folding. And I told you how important protein folding is. So when we inhibit protein folding, these animals can really not survive. So all we get are these dead corpses. But when we have mutants which have improved protein quality, they can actually survive this toxic drug, and we can actually get survivors. Now we asked if these survivors are also long-lived, and we indeed found that lots of these resistant animals were then also long-lived. So this means already that perhaps by improving protein quality control, we can extend lifespan. We then used a method to analyze the whole genome of these animals, a method that is very similar to the way that our own human genome was sequenced about 10 or more than 10 years ago. And we actually sequenced about 24 of these worm genomes. And one gene that came out of this analysis over and over and over again is a gene that is responsible for making a certain enzyme. And this is the enzyme that provides the precursors for the glue that's required for protein folding. So just to recap, what we have in our long-lived animals is an activation of the enzyme that produces the glue molecules required for protein folding. This was important for us because, first of all, this enzyme had never before been associated with aging, so we think we actually discovered a new longevity gene. But secondly, and I think much more important, we have almost the same enzyme in our cells. So we think that here in this case we actually were able to learn something from this very simple model organism about our own biology that might be relevant for human health as well. Now, I told you about the longevity in the worm, and what we can also model in the worm is age-related diseases. So we can actually give certain genes to worms that are responsible in humans for causing age-related diseases. So here's a worm which expresses a protein that causes Parkinson's disease. And as you can see, it can hardly move. Now it moves only the head. So Parkinson's disease is a progressive neurodegenerative disease and leads to massive paralysis. And this we can really model in these worms. Now, when we provide these animals with the precursors for these glue molecules that improve protein folding, this paralysis is almost gone, and we see great improvement in their motility. Once they begin moving. Yes. Okay, so treatment, no treatment. These are the same worms. They both have the Parkinson's gene, but when they're treated with this precursor for this glue, they actually have much better mobility. So this means that here we have an intervention which not only extends the lifespan of these worms by extending their old age, but it really seems to slow the aging process throughout their life and to give them a longer healthy span of life. And this is what's really important here, because what we're really addressing, as I said, is age-associated diseases. So this treatment actually prevents these age-associated diseases. So there are two things that I wanted to say today. One is that we can still find new longevity genes by interrogating the genome in an unbiased way. And this story shows that there's probably a lot more genes out there that we can find by doing this. The other point is a little bit more fundamental. The other point is that aging, and this story shows this again, is not a process of random decay. If aging was a process of random decay, then mutating single genes would not be able to slow the all-over decline in a whole organism as aging occurs. But since single gene mutations are actually able to extend lifespan of the whole organism, this means that aging has a regulated component. And if aging has a regulated component, then we might be able to understand this regulated component and perhaps manipulate it in such a way that we can also improve our own healthy span of living. So, aging researchers have used simple model organisms such as worms and flies and yeast, and also in mice. We have learned a lot about pathways and interventions that can extend lifespan. And if you were now to ask me what you can do now to extend your lifespan, then I would ask you to wait a little bit, because we're still busy at translating all these findings that we have in simple model organisms into the human system. And I'm pretty convinced that during my own lifetime, I'm going to see this research really play out in a beneficial way, and that we can actually find ways to improve health in humans as well. But until then, I would recommend that you stay healthy and probably do what your mother's already told you, which is don't stress out, get enough sleep, have friends, laugh a lot, don't drink too much, don't smoke. And your mother's probably didn't tell you this one. Appreciate the molecular maintenance machines that keep you going. Thank you so much.